Hello again, this is Dr. Carver, and you are going again to travel with me in my journey discovering those topics that cover your 1409. Today, we are going to talk about the immune system and the lymphatic system. Okay, so an overview of the immune system. The immune system, I mean, fight infections and cancers, and the lymphatic system is helping the immune system, is a part of the immune system. So we are going to do what is lymph before we go any further. Lymph is just a watery fluid that bathes tissue and organs, and that contain protective cells um, that we call white cells, white blood cells, or erythrocyte. It doesn't contain red blood cells or erythrocyte. The lymph move the body through all the body. I mean, it move through the lymphatic system, which is made of vessels, lymphatic duct, as you can see over here, those lymphatic duct. And also we have some lymphatic glands and organs, such as thymus, spleen, a red blood, red bone marrow. We do have also the tonsils. The adenoids are also organs, lymphatic organs. Although the immune system is characterized by circulating cells through the body, the regulations, maturations, and intercommunications of immune factors happen at specific sites. The blood circulates immune cells, protein, and other factors through the body. You know how many white cells circulate in the blood? Most of the blood cells that circulate in the blood are red blood cells, are erythrocytes. Only 0.1%, 0.1% of the blood cells are leukocytes. The thing is that all the cells of the immune system can travel between, and we are going to see later on in the table all those immune cells can travel between the distinct lymphatic and blood circulatory system. The lymphatic and the blood circulatory system are separated by what we call interstitial space. So, just to explain it a little more, the immune system, it came from where? It came from what? It came from actually stem embryonic cells in the bone marrow. And it's so funny how when we were kids, we have all our bones contains bone marrow, red bone marrow. When we become aged, those are going to be reduced to the sternum, reduced to the skulls. And this is why our immunity system also go down. So let me go back. So let's recall that all the cells of the immune system originate from stem cells in the bone marrow. B cells, one of the immune cells. T cells is one of the immune cells. We will look at the tables. DR matures happen in the bone marrow, the B and T. They are originated in the bone marrow and mature for B in the bone marrow, but for T, even if it's originate from bone marrow, you will mature into, I will call it naive T cells in the thymus. And after that, the B and the T cells are going to circulate to their various destinations, doing their immune work. The lymph nodes that you are seeing over here, they are all over the body, you see it, scattered through all the body. You know that the way I talk about B and T cells over here, because they are going to large house a large population of T and B cells. In those lymph nodes, we don't find only large amount of T and B, we find also some dendritic cells and some macrophages. The lymph then gather, you see, the lymph is going to gather antigens or those other version, little version antigens is a pathogen, um, and drain it from a tissue. And then those antigens are filtered through the lymph nodes. And what we have in those lymph nodes, we have those immune cells that they are going to destroy those antigens. And after they filter this lymph node and get rid of all those antigens in those nodes, the lymph is then returned to the circulations. In this illustration, as you can see some of the lymphatic organ visible, the thymus, the spleen, and the red bone marrow. Show also the lymph node that they are connected by the lymphatic vessels. In the next illustration, you just see a bigger size or has been magnified, enlarged the lymph node. So you can see that how the lymph node enter, the interstitial fluid enter through the lymphatic vessels from one area, and then inside of the lymph node, he got filtered, destroy all the pathogen, and then leave the lymph node via another lymphatic vessel on the other end. So here, what we have, I'm sorry, sometimes my microphone just go away and I don't know which I was. So in this slide, I was just covering, as I said, all the cells of the immune systems. The macrophage do phagocytosis. The B cells get developed and matures in the bone marrow cells, and they also develop. They are the one that produce antibodies, and not only that, they develop memory cells. The T lymphocyte cells, they are two types: toxic T cells and helper. I will come back and develop each type of cells, and I come back to this summarize the tables over here. The lymphatic organs, we already cover it very nicely, but doesn't hurt to go over it again. So as I said, the immune system get reduced to age, and the thymus is larger in the children. He shrink when we age. And you remember, the thymus is where the lymphocyte T cells got mature. The lymph node, they are small. I already cover it very nicely, and even we have enlarged pictures of it, illustrations of it. The first thing when you have any infections like for example, bronchiolitis, you went to the doctors. The first thing he look at is under your ear to see if they have any swollen lymph nodes. That indicates that your body is having an infections and is trying to fight these infections. We already cover the spleen, which is one of the lymphatic organs. He filtered the blood. We can survive with no spleen, but our immunity system is reduced, and therefore we are susceptible to infections easier. So the vertebrate, we have two parts of a immune system. We have the innate immune system. Innate is actually not really specific. Like our body, for example, we saw that he fight pathogens, but do we know exactly there are variety of pathogens? It's not specific. It's not like the skin is going to fight only this kind of pathogen. He fight any kind of pathogens. So this is a physical barrier, like the skin, the hair, the cilia, even the hair that we have in our nose, for example. He does a fight because he filters the air that is coming inside our body. The mucous membrane, the mucus and chemical secretions, the enzymes that exist in our mouth, the digestive enzymes, the acidity of the stomach. He destroys some bacteria that they are on some pathogen that they are coming with the food there. Internal defense, not limited to the inflammatory response, but also productions of competent proteins, stimulations of phagocytosis, and stimulations of natural that involve also some natural killer cells that they are part of immune system. And then we came to the adaptive immune system. This is very highly specific. Why it's specific? Because they develop antibodies against a specific antigen. That's one. They do what we call cell-mediated immune response. I mean, this is not the cell that will go to destroy this pathogen. It's this cell. So it's cell-mediated immune response. And they do memory response. We will cover all this while I am going through our journey in this. The cells, the body have immune cells, in the immune system, and this is what giving him the capability to fight. and reject, repel, trash all foreign substance, pathogens, and cancer cells. The non-specific immune system doesn't do any discriminations. We don't know exactly. You can fight anything. The specific, it's a specific antigen. Antigen, it's kind of like a variant of a pathogen that stimulate the immune system. Non-specific or innate immune system, I said the first line of the defense is the barrier to enter, so physical barrier. Then we develop the after that we develop the inflammatory response, producing competent complement proteins, and the NK cells are stimulated. The barrier to enter can be mechanical, such as the skin, and their mucus. The skin also have some um, sweat glands, oils, and those sweat glands are acidic enough to inhibit the invasions by the bacteria. The skin have several layers. You have the epidermis, you have the dermis where those sweat glands exist, and we have the hypodermis. Hypo is the last layer. HYTH. Hypodermis. Let me write it down better than talking because I got hypo. Dermis. The hypodermis is the last one. You see where this fat exists? That's the hypodermis, where those fatty that appears yellow. That's an hypodermis. That's the dermis, this blue thing that you are seeing over here. This is where the sweat glands, and this is the epidermis. This is where those hair shafts came out. Notice that the hair shaft that they are coming from the dermis, right? But they are coming outside, showing up in the epidermis. And the sweat glands are also found in the dermis, but they have opening on the surface of the skin. So those sweat glands are acidic enough to, as I said, to fight against any invasions. Again, we have also those the upper tract. They are lined with the ciliated cells that sweep mucus and trap particles that they are coming through the airways. The stomach also have, you remember the pH is between 1.5 to 2.5 pH. It's tremendously acidic to inhibit or kill any bacteria. Not even that, they have what we call a good bacteria that they exist in our flora intestinal and also in some vagina. This is why we really suggest to our, since we were kid, to not use highly soap. There are special soap that have to be neutral soap to clean our intimate area, because if you destroy the pH of your vagina, you are going to destroy those pH by destroying the by changing the pH of your vagina. You are destroying those bacteria that they are protecting actually your vagina from any invasions, foreign invasions. Okay, and this is why we have to eat a lot of probiotic products that give us healthy bacteria to keep our intestinal flora functionings correctly. One of those innate we are over here. Remember this table? We talk about all this part. Now we are at the inflammatory. We are at the internal defense. The inflammatory. Zero of event happens actually whenever the tissue is damaged. So the first cytokine to be produced encourage inflammations. How I know inflammations? Because we can notice a localized redness, swelling, and the part where your skin has been cut, it starts to be like hot, right? It's a heat and pain. This is all you can visualize and you can sense. Inflammation, what is it? Inflammation is a response to physical trauma, such as a cut, as I said, or chemical irritations, and infections by viruses, bacteria, or fungi, by any pathogens. What are the chemicals that trigger these inflammations? Well, there are some chemical signals that this trigger an inflammatory response and cause those inflammations. So those chemical signals, they do some inflammatory, enter the extracellular fluid, and what they do, they are going to expand, dilate the capillary. Oh, once the capillary is completely dilated, you know what's happened? The capillary walls become more permeable. Oops. So he become more permeable. That means is leaking over here. He become leaky. The serum and other compound leaking from the capillary, they are the one that they are going to cause the swelling of the area, which in turn cause a pain. It's like a chemotactisms now. Various kind of white blood cells are attracted to the area inflammatory. We talk about each one of them right now, and we can see them in these illustrations. Neutrophils. Wow. For example, neutrophils is the first one coming. This neutrophil is the first one coming and engulf and digest the pathogen. This neutrophil. They are the most abundant, by the way, the neutrophils, white cells of the human system. Next, who came? The neutrophil is coming. Macrophage are the next. They follow the neutrophil and take over the phagocytosis functions. The neutrophil started, right? They are eating. The macrophages came. Hey, they are eating more. Macrophages are the next. So they take over the phagocytosis functions, and therefore they are involved in the resolutions of this inflamed site. So they try to clean up cells, debris, and pathogens. Who are the next? Those little cytokines. What they do? The cytokines, they are going to send the feedback to the nerve system to bring about the overall symptoms of feeling sick, which include muscles pain, nausea, and they are also going to increase, you are going also to feel fever. This is why sometimes when we have an infections and you feel a fever, it's um, it's not something very bad. Actually, it's good. That means your body is fighting those pathogens. So this is why, for those reasons, I mean, the cytokines that increase the core body temperature causing fever, the elevated temperature of fever inhibit actually the growth of the pathogen and speed up the cellular repair process. So for those reasons, I mean, the suppression of of a fever is not a solution over here because hear me out, if this fever is not dangerously high, okay? So let's summarize. So these illustrations that I have over here show an inflammatory response. They show cross sections of the skin, epidermis followed by the dermal. On the dermal, this is where the most of the capillaries and blood vessels exist. We have a cath, and we have an innate defense over here. It's followed by the skin and the mucous membrane, which are the physical barrier for the entry. But unfortunately, capillaries, skin is cut, the capillaries is leaking. Pathogen enter the body and attach themselves to dendrite cells. The capillaries have complement proteins and interference in the plasma. The natural cells in the capillary release monocyte, which becomes a macrophage and neutrophils. Neutrophils start this phagocytosis process, finished by the macrophage. Cytokinosis help also, as I said, by attacking those pathogens because they also increase the core of the body temperature. They do like a feedback, bringing about the overall feeling sick and feeling fevers. Those it's because the cytokine is fighting back. Okay, guys. What are those? We hear the word complement proteins that they are part also of the immune. Cells, what are they? There are about 20 type of proteins called complement system. They are also activated by infections or just by the activity of the cells of the adaptive immune system. And their function is to destroy any extracellular pathogens. Liver cells, macrophage cells, synthesize inactive forms of complement protein continuously. Liver cells, liver work very hard, really. Liver cells and macrophage cells synthesize inactive form of those complement proteins continuously. Those proteins are abundant in the blood serums and are able of responding immediately to infections microorganisms. So they are very abundant and they give immediate response to any micro infections by microorganisms. Why we call it complement? Because it just for the some reason he complete the innate and adaptive immune system function. He completed. So we call it complement system. They are proteins, all right? How they complete that? They are going to bound to the surface of microorganisms. They will adapt to their system. We design those complement proteins by big C and subscribe. Big C1, big C2. So the complement, the C-bound complement proteins, we know we call it a C, big C. The big C bend to the surface of microorganisms and are particularly attracted to pathogens that are already tagged by the adaptive immune system. So they are going to complete what the immune system already tagged this microorganism, and they are going to complete these functions. The taggings involve this attachment of a specific proteins that we call antibodies. We will discuss that later to the pathogen. When they attach this complement where the antibody is there already by the immune system, the antibodies change shape, providing a binding site for one of the complements. It's beautiful, this immune system, how it's work. I promise you that sometimes I just been amazed looking and illustrating this. So first, the immune system, the adaptive immune system, is trying to destroy the microorganism. They have antibodies. The complement come and attach. The antibodies, as soon as the complement attach, change the shape, providing a bonding site for the complement. Hey, came with me. This is exactly what they are doing. Came with me, my body. The antibodies, they change the shape, providing a bonding, they provide now a bonding site to the complement. After the first few complements bound, a cascade of bonding in a specific sequence of proteins follow, in which the pathogen rapidly becomes coated in complement proteins. The pathogen is completely circulated now, completely is a prisoner. It's amazing, isn't it? Complement proteins, big C, we call it big C, right? Perform several functions. One of which is to serve as a marker to indicate the presence of pathogen to phagocytosis cells. Look at what they do. They are not only helping the antibodies, but they are, once they circulate, the phagocytosis cells come. It's a signal. They are going to come and enhance the engulfment. Some complement proteins, they can combine to open a pore, they combine between them and actually open a pore. And it's like trying to destroy the microbial cell membrane, and they will. They open a pore in the microbial cell membrane. And when the membrane is not there, the cell, the bad cells, the microbial cells, is lysed, causing a lysis of the microbial cell. It doesn't hurt again to read this, right? To read this slide, but I did explain it very well. Another thing that I didn't talk about it, that those C proteins can release more white blood cells, primary neutrophils, primary neutrophils, because they are going to stimulate the phagocytosis. And this is to show that they combine with each other to lyse actually the plasma membrane of the pathogen. It make a holes, pores on it to destroy the plasma membrane. All right, young people, let's move on. We talk about the complement. Now we are at the natural NK cells. In this illustration, you see that the natural K cells are such as monocyte, macrophage, and neutrophil. Those are the histology of those cells, how they look like. The NK, what are they? It's a lymphocyte also cells. It's a part cells of the immune cells system. It's a white blood cells that contain a large nucleus. You can see it is the most large nucleus is in the NK cells. Most of the lymphocytes They are associated by the adaptive immune response. However, the infected cells can be identified and destroyed by the NK. The NK is the only lymphocyte of the innate immune system. If I go back to my table that is in slide 9, no, not this one. I think it's slide 9. Is the only lymphocyte is the NK of the innate internal as internal defense. Go back to my slide. They don't have any memories of previous contact. They don't have it, any memories. The only lymphocyte that do memories are B cells, anyway. The natural NK cells is a lymphocyte that can kill cells infected with viruses or cancerous cells. And not only that, they can also identify the infections, especially from viruses. Because those viruses are we are going to see HIV virus and our HIV virus expression because they alter the major histocompatibility complex, the MHC molecules on the surface of the infected cells. What are those MHC cells? Are proteins on the surface of all nucleated cells which provide samples of the cell's internal environment at any given time. And healthy cells, either they are infected cancerous, they develop an alterated, we call it histocompatibility complex class 1 on their surface. So therefore, they will recognize the NK, they will recognize those cells that they have an alterated MHC class 1 complex. Because the cells that is being infected, he will develop, it's like a flag in their surface that they are being infected. After the NK cells detect an infected or tumor cells, it induces a programmed cell death or apoptosis that we call. The cell, therefore, this is the killer, the NK, is going to program the cell that he recognized to death. But not any death. It's not like necrosis, archaic death. It's a death that involves a lot of signaling process. A lot of molecules are involved. We call that a programmed cell death or apoptosis. Then after that, phagocytic, let's speak English correctly because I always say those scientific words in French. Phagocytic cells then came along and digest the cell debris left behind. So the NK cells, they do what we call the same method as the T cells to kill the cells. The only difference, it's not specific. They don't increase in number when they are exposed to an antigen. So they don't have any memory or any previous contact before. The natural killer cells, the NK cells, are constantly patrolling the body and are an effective mechanism for controlling potential infection and preventing cancer progressions. Here in this cells, we just show the various immune cells histology in these illustrations over. Next, we talk about adaptive immunity, and I want to know, and we talk about antigen. Even if I explain it, I want to go back to that. All right. The adaptive mean acquire immune system, take days or even weeks to become established. My organism is adapted to always fight this pathogen if it's around. So therefore, it's much longer than the innate response. However, the adaptive immunity is more specific to an inviting pathogen. So adaptive immunity is an immunity that occurs after exposure to an antigen either from a pathogen or vaccinations. What is an antigen? Antigen is a molecule that stimulates a response in the immune system. It's a part of pathogen, foreign cells or cancer cells, an antigen. It's a small variant of a pathogen. It's a molecule. So the antigen is a molecule that stimulates a response to a human system. And this part of immune system is activated when the innate response is insufficient to control an infections. Therefore, in fact, without informations from the innate immune system, the adaptive system cannot be mobilized. And we have two types of adaptive system. Go back to our slide number 9. We have two types. We have one that they are mediated immune response, the cell mediated, and the other one that they are humoral immune response. The one that is cell mediated response, it's controlled by activated T cells. The one that is humoral immune is activated by the B cells and antibodies. Activated T and B cells, um, they are going to bind to the plasma membrane, right? Special receptors, they are like lock and key. So those activated T and B cells are binding site are specific to the molecule on the pathogen. Their attack, of course, will kill the pathogen directly, or they can just secrete antibodies that enhance the phagocytosis of the pathogen and disturb the infections. The adaptive immune system also involves memory to give the host long-term protections from reinfections with the same type of pathogen. On re-exposure, this host memory will facilitate a rapid and a powerful power. think in general, I cover everything that this is in this slide. We already talked where the T cells get matures. They don't produce antibodies. The B cells produce the antibodies, doing the humoral immune response. They always, as I said, the adaptive immune also involve a memory to give a host a long-term protections. Like when we receive recover from measles, we are not going to get it again another time because our body is already know how to fight it. This is why you don't feel it anymore. We are going to next slide. The B Let me finish with the T cells. And by talking about B, I want to talk about why I am talking about B cells. I will also go back to the T cells. The lymphocytes, which are blood cells, they are white blood cells. They are formed in the red bone marrow, right? And they are can be found in many flat bones, such as sternum, shoulder, pelvic bones. We already said it that the two type of lymphocyte of adaptive immune response are B and C and T cells. Whether an immature lymphocyte B or become a B or T depends actually where in the body it got mature. The B is got mature in bone marrow. This is why we call it a B, by the way. This is why we name it a B for bone marrow. The T, even if it got formed in the bone marrow, it will migrate to the thymus where they get matures. So here we call it a T because it got mature in the thymus. THYMUS. Just want to emphasize this part. Maturation of a B or T cells becoming immunocompetent. What's that mean? That means that they can recognize by binding a specific molecular antigen during the maturation process. Immunocompetence. B and T cells that bind too strongly to the body's own cells are eliminated in order to minimize an immune response against the body's own tissue. Self. You see the word self in some of those slides. This is what's mean. Those cells that react weakly to the body's own cells but have highly specific receptors on their cell surface that allow them to recognize a foreign molecule or antigens. Let me make it clear. During the maturation process, there are some cells, B and T, either in the bone marrow for B or either in the thymus for T, they become too attached to their own body, their own cells. that they can cause what we call immunocompetent. And they develop a response against the body's own tissue, their own body. They have to be eliminated in order to minimize this immune response against the body's own tissue. The other cells that react weakly to the body's own cells have highly specific receptors on their cell surface that allow them to recognize a foreign molecule, but not their body, their own body. They have specific receptors. This process happens during the fetal life development and continue through the adult life. And the specificity of this receptor is determined, actually, its genetics is determined by the genetics of the individual. And it's present before a foreign molecule is introduced to the body. So it's dang, isn't it? It's the genetics and not the exposure to the pathogen or to the microorganism that initially provide a vast array of cells able of binding to different specific foreign molecules. Once they become, once they become immunocompetent, the T and B cells will migrate to the spleen and lymph node where they will remain until they are cold during an infection. So to become immunocompetent, look at how the process they do. First, we have to get rid of those who, during the maturation process, they bend too strongly to the body own cells that they destroy it. And those that react weakly to the body's own cells, even they have highly specific receptors on there that allow them to recognize a foreign antigen. Where those highly specific receptor came, it's genetics. It's genetics. It's from your genes. So the B cells, we explain it. It's involved in humoral immune response, which targets pathogens in blood and lymph. And the T cells are involved in the cell-mediated immune response, which targets infected cells. Let's continue. Um, each B cells have only one kind of antigen receptors, which make every B cells different from each other. when a B cells meet with an antigen and bind to its receptors, the antigen molecule is brought into the cells by endocytosis. When a B cells he is bonding to the antigen via the specific receptor, the antigen molecules came inside by endocytosis and reappear after that in the surface of the cells through what we call the MHC class 2. The major histocompatibility complex class 2 molecules. When this process is finished, when I have the MHC class 2 molecule in the top now of the cells that has been infected, the B cells is sensitized. In most cases, the sensitized B cells encounters a specific kind of T cells to help her. We call it exactly helper T cells. The T cells that's already been there, it's already been activated, by the way, by this MHC class 2. And use a release of cytokine. Hear me out. So the B cells came in, get by specific receptor the antigen. Antigen is inside. The cells will show MHC class 2 molecules. At this moment, T helper came in to help the B cells. I will induce a release of cytokine. Those cytokines are going to induce the B cells to divide rapidly, which makes thousands of identical B cells. Those daughter cells that has been made from the original B cells become either plasma cells or memory B cells. The memory B cells remain inactive at this point until another later encore with the antigen caused by reactions by the same bacteria or viruses results in them dividing into new populations of plasma cells and etc. The plasma cells, on the other hand, produce and secrete large quantity up to 100 millions molecules per hour of antibody molecule. An antibody is also known as hemoglobin. It's a protein that is produced by the plasma cells after stimulations or after infections by an antigen. The antibodies are the key. They are the key agents of humoral immunity. They are happening in the blood. We find them also in the gastric and mucous secretion, and even this is why we always ask to breastfeed the babies. The antibodies in these bodily fluids can be bound pathogen and mark them for destructions by pathogenesis. So in these illustrations, that show B cells and the antibody response, it show pathogen that develop antigens in its surface. All right, they are those small molecules that it's just a small variant of this pathogen. They are going to bind to the B receptor, the BCR, that fit the antigen surrounding, and then I explained to you how the cytokines are going to be produced. I did talk about it. So they are going actually when they band. This illustration is making it very, very simple, but it's more complicated than that. Actually, when they came, the virus, the pathogen, the antigen is inside by endocytosis. The cells that is being infected that have those specific uh, receptors in the top B cells, they are going to develop class 2 the major histocompatibility class 2 molecules, and the B cells is then sensitized, and the helper T cells are coming to rescue that process that is happening, is to help this process. And when in doing that, is going to stimulate the productions of cytokine. And those cytokine, what they are going to do, they will release more. They will stimulate the B cells to divide more. So the B cells is going to divide into more cells, and they will divide into memory B cells. That's what is this illustration is about. Next, oh, we already talked about that. I already explained all this part. So we move to the next one, which is the antibody response. We barely said that the antibodies, the immunoglobulins, are the antibodies that they are able of combining with specific antigen. They circulate in the blood, lymphatic system, and bend with the antigen even whenever he found it. The bacteria can fight infections in several ways. They can be chemical interactions. They can create a bridge between different particles containing antigenic sites, clumping them together and preventing their proper functionals. The antigen antibodies complex stimulate the complement system that we described earlier, uh, the C system that will destroy the cells bearing the antigens. They can also have some phagocytic cells. They can also be attracted by this antigen antibodies complex, and phagocytosis process can start. The antibodies The antibodies can, sorry, can also mark pathogens for destructions by phagocytosis, such as macrophage or neutrophil, in a process called opsonizations. In a process called complement fixation, some antibodies provide a place for the complement protein to bind. I already explained that when I talk about the C complex. The combination of the antibody and the C complex promotes rapid clearing of the pathogen, rapid destructions of the pathogen. So the predictions, by the way, the predictions of the antibody, again, I repeat it again, a response of the plasma cells in response to an antigen, this is what we call active immunity. There is also, this is an active immunity, right? Because our cells produce those antibodies. We have also a passive immune response, where those antibodies came from our outside source instead of the individual own plasma cell and are introduced in the host. For example, antibodies circulating in pregnant person's body move across the placenta into the developing fetus. The child here benefits from the presence of these antibodies for several months after birth. During COVID, we did give some immunoglobulin injections, IV, to those patients suffering from COVID coming from their relatives. This is a passive immune response. It's not the body that developed those antibodies, but it's coming from outside. And we do it also to fight hepatitis infections. We inject antibodies in the blood serum. This gives immediate protection since the body does not need the time required amount for its own response. We arrive at the slide 27, and this is show just the B receptors that they are embedded in the membrane of a B cells and band a variety of antigen to their variable regions. Not all the circulating antigens band, have um, What I am saying, it's an antigen, just to remind you, is actually a molecule that stimulates a response in human systems, all right? Not every molecule is an antigen. The B cells participate in response to antigen presence by producing, by actually doing humoral immunity by producing antibodies that circulate through the body and bind the antigen whenever it's there. This is also known, as I said, humoral immune response. As we discussed it earlier, during maturations of B cells, a set of highly specific B cells are produced that have many antigen receptor molecules in their membrane. Here, example of EBO blood type cells. I love to see it in the tables. I will summarize it. So the blue type A, or which antigen we have in the blood, they are an antigen A. Therefore, they cannot have an antibody anti-A because they will destroy the A. They have an antibody anti-B. So therefore, they cannot receive a B because B have an antigen B. They cannot receive B. They can receive only A. B, the same things. They have antigen in the surface of the B cells is a B. They cannot have circulating antibody anti-A. They cannot have anti-B because they will destroy it. They will have an anti-A. So therefore, they cannot receive in A. They can receive only B. I will come back to O. A and B, they have both. They have antigen A, they have antigen B. So therefore, they cannot have antibodies A, they cannot have antibodies B. Because if they have the antibody A, they will destroy A. If he have an antibody B, he will destroy B. So he cannot have both of them. So therefore, he can receive all those blood, A, B, and AB. Oh, what is it? It's the universal donor. We know that because he doesn't have any antigen. And therefore, the antibodies in their plasma, they have antibodies A and antibodies B. Compatible donor is only A. We're arriving to the T cells. Even if I talk more about it, T cells have many functions. Some respond to, I mean, they get matures in the thymus. Only 5% leave the thymus. They have unique also receptors. We call that TCR. I mean, not all the T cells have the same specific receptors. Unlike B cells, the T cells cannot recognize an antigen without the help of B. The antigen have to be present them via what we call antigen presenting cells, the APC. And some of those APCs, what they do, they release cytokines. Some of those stimulus is when the antibody is being secreted by B. And we talk about this major complex, major histocompatibility complex 1 and 2, that when a virus is infected in the cell what is do, he produce on the surface this MHC, moving the cells to be presented to the T to come and help, I mean destroy it. How many T cells? When these T cells got stimulated, they produce two types of cells. Cytotoxic T cells and helper T cells. When the infections wanes, the T cells can undergo those programmed cell death. That's called. So in this illustration, what you saw, it show an activation of the T cells. It show virus entering a macrophage. The APC then is inside of lysosome. The macrophage has many those MHCC, the major histocompatibility complex proteins and antigen from the virus, which are located on the surface of the macrophage as MHC2 plus antigen complex, EPC. So then the T is being stimulated. The T helpers came in with its a specific receptor TCR, and it's fit with the measure of, you can see over here, in this part, the TCR and the T helpers fit. So the T helper is then activated, and therefore release those cytokines that will help destroying this pathogen. You know, those cytokines can stimulate the B cells to produce, to divide and be memory B cells. So as I said, there are two main type of T cells: T helpers, T lymphocyte helper, TH, and cytotoxic T lymphocyte. The T helper functions indirectly to tell other immune system about the potential pathogen. He's a helper. He recognizes specific antigen present by those class 2 MHC class 2 complex of APC, of the antigen presenting complex. They have two populations. I'm not going to enter on this. Of the T helpers, we have populations number 1 and we have populations number 2. But let's just take it that they are going to secrete cytokine when once the T helpers got stimulated, you will secrete cytokines, as I said earlier, to enhance this activity of macrophage. And not only that, they will also, and this is part of the T helper type 2 cells, helper Th2, they are going to stimulate the naive B cells, the one that they didn't sensitize yet, antibodies, to sensitize antibodies. So the T helpers are doing two functions. One function secretes cytokines to enhance the mechanism of phagocytosis, and the other one is to stimulate the naive B cells to produce antibodies. We will talk about it when we talk about the HEV infections. The cytotoxic T cells, they are the key compound of the cell-mediated part of the adaptive immune system. Remember, the T cells, they do what we call mediated immunity. All right, all right. So they are the key player of the cell-mediated part of the adaptive immune system. They can attack and destroy the infected cells, are particularly very important in protecting against, therefore, against, they are very important for protecting because they play a major role. I will explain why. Because when a virus replicates within cells where they are shielded from extracellular contact with circulating antibodies. Look at that. We have antibodies, but they are hiding. They are inside of the cells, and oh, they have their freedom. They start replicating. This is why the T cells are very important, because they are the one responsible for this response to virus infections. It's not when you have virus infections, therefore antibodies is coming in from the B cells. No, the cytotoxic T cells is the one that will raise the flag. It's the one that once activated, it creates a large clone of cells with one specific set of cell surface. So the T cells has ability to destroy the infected cells before even the pathogen can replicate and escape. And therefore halting the progression of the intracellular infections. They are also supporting not only B cells to produce antibodies, but they also support NK cells, the natural killers, to destroy early cancers. The cytokines that they are being secreted by the T helper response that stimulate macrophage also stimulate the T cells and enhance their ability to identify and destroy infected cells and tumors. In general, I mean the plasma cells, B cells, and the T cells, they are collectively effective because they are involved in affecting the immune response to kill pathogens in an infected host cells. In these illustrations, this is an electromicrograms illustrations in your left. Um, it show T cells, large cytotic T cells, in like a blue. And in the left, you have an illustration of T cells that attack a target cells. The T cells, as you can see, they have some perforin, some enzymes inside the vesicles. We call that granizymes in those vesicles. They attack the target cells. The T cells has a receptors that will form an histocompatibility, a major histocompatibility to plus the antigen that is coming from the host cells, the target cells. The vesicle will perform actually it will stick to the target cells and form a pore. And therefore, when those vesicles that I have over here, this is what we call them performing vesicles, they are going to be in contact. This is just a large part of the plasma membrane of the target cells. That is my target cells. This is actually my target cells, the same one over here. This is just the membrane. He came in contact with this membrane, all right? And he formed like a pore. He performed. This is why we call it vesicle perforing. And once he performed this target cells membrane, he will form those pores. The granzymes that exist in those cytotoxic C, those enzymes are going to come inside of the target cells through the pore, resulting in apoptosis, causing this target cell to die. The program the cell death. So this is just actually summaries of all what we said. Characteristic of the T cells, they are produced, they are responsible, very effective in cellular response to virus infections and cancer cells. They don't produce antibodies, they are produced in the bone marrow by gut matures in the thymus. This is why we give them the letter T. They can the histocompatibility, the antigen must be present to a T cells in a groove of an MHC class 1 protein. Cytotoxic C destroyed non-self antigen bearing cells and the helper secrete cytokine that control the immune response. And the T helpers here, some things that is not covered, they have also, they can stimulate the they stimulate the naive B cells to secrete antibodies. This is tissue transplant and immune response. This is an example that certain organ could be transplanted easily from one person to another if the body do not reject them. Rejections can happen because the T cells and antibodies bring about the destructions because they will consider it, and it is a foreign tissue coming inside of the body. So the organ rejections can be controlled by carefully selecting the donor and by injecting also administration and immunosuppressive drug to not let the inside immune system destroying this foreign body coming inside of the receptor body. So this is why the first thing we look at for the any organ donor is to see the same type of image AC. A lot of people are registered as organ donor. I am one of them in my driver license. So yeah, this is to save their life. All right. Some drug works by inhibiting response T cells to cytokine to weaken actually the immune receptor, so the acceptor can accept the donor organ or tissue. Immunizations, vaccines are substances that usually do not cause illness, but cause the immune system to respond to them. So in another word, you stimulate the immune system and you develop therefore this memory antibodies by the T cells, by the B cells. Oops, oh God, I said something wrong here. It's not the T cells that produce antibodies, right? It's the B cells. The pathogen or their products that have been treated, they are not virulent anymore. The protein produced by genetically engineered bacteria. This is what they contain, those immunizations, those vaccines. They can be active or passive as immunity. Active immunity, even if I talk about it, is it's promoted by immunizations. After a vaccine is given, antibody titer in blood can be measured. Primary response is the first response to this antigen. Secondary response is faster. It produces more antibodies. This is why we use what we call booster dose, because we are boosting and we are going to keep this quantity of antibodies big enough in our body. Usually longer lasting, but as I said, the booster might be required after several, they said several years. For COVID, I mean for this coronavirus, we require every 3 months, every 6 months, because of the mutations of this virus and his degree of virality. Um, have actually plot the primary and secondary response to of an active immunity due to immunizations. the graph plot plasma antibody concentration on the vectorial axis, and the time in days from 0 to 100 in the x-axis per days. The first response to the vaccine is at day 0. The second response to the body is at day 60, with the concentration of the plasma antibody higher on day, if you look at the peak, is in it's in day 90. So if you look at the second exposure to the vaccines, is giving me better antibodies concentrations. The passive immunity prepared antibodies. This is what I said, immunoglobulin, that not produced by the individual's plasma, but we can have them from another person. It's temporary. They don't produce any memory cells. Newborn, I went through this, receiving antibodies from their mom across the placenta, and through also the breastfeeding. It only lasts few months. So recently, we did give injections of the uh, uh, prepare hemoglobin, prepare antibodies to given COVID patients, and to also patients suffering from hepatitis disorder. Um, they have some disorders for immune systems. The immune systems, cannot distinguish self from non-self. Allergy, for example. Hypersensitivity to substances that ordinarily would not cause an immune response. Pollen, food, animal hair, allergen, food allergy. I do have very strong allergy against peanut. It's regulated by cytokines secreted by both T and macrophage. Yeah, the season of pollen. Immediate allergic response caused by receptor or mast cells on the tissue. Allergens trigger cause release histamine and can happen within a second of exposure. This is why as soon as you're in contact, you start sneezing. Symptom can vary by extreme of an anaphylactic shock, sudden life-threatening drop in blood pressures. Me, I swallow, I become very big, large, at the point like I cannot even open my eyes. It was very scary the first time when I got it. Or delayed allergen response. This is initiated by, we call it memory T cells at the site of allergen contact in the body. The skin test for tuberculosis. It's a passive test is a redness at the site. It shows previous exposure to the tuberculosis when it's answered. That means you've been exposed to the TB. But the thing is, I'm going to tell you this because us in France, we receive a vaccine against the tuberculosis. I know here in United States, we did done it. So the first things I remember, because I was working in the lab, they done this TB test and he got positive. And he said, you've been in contact with tuberculosis. I said, no, I never have it. Then after that, I got a smart doctor. They said, are you being vaccinated? Where are you from? I said, I'm coming from France. He said, oh, you got vaccinated. This is why. Some develop also dermatalized skin when we talk, when they are in contact of some, even jewelry, some cosmetics, skin allergy. They develop also some fungus in their skin when they are allergic to dogs or cats. Autoimmune disease. Autoimmune disease when TC cells or antibodies attack the body own cells. The cause is not really known. We have this autoimmune disease called neuromuscular junctions that do not work properly, and therefore, if they don't work properly, they weakens the muscles contractions. We call that disease the myasthenia gravis. The multiple sclerosis, MS, the myelin, this a protective sheet that exists around nerve fibers. Not all the nerve fibers have this myelin, but most of them have it. They cause several neuromuscular disorder. They can break down, causing several neuromuscular disorder. Kidney damage, also. Arthritis, it's an autoimmune disease. Look at this arthritis over here. Sad, isn't it? Ah, God. HIV, autoimmune immunodeficient syndrome. And HIV, it's a human immunodeficient virus. He live and destroyed T cells. Body is able to maintain adequate T cells for some times, but without therapy, the number of T cells can drop. The HIV is, it can drop. The person is unable to fight the virus, any viruses. He become immunodeficient. He is going to be subject to any virus, fungi, bacteria that in our daily matter we fight. Those poor people cannot fight it anymore. It can be transmitted sexually, needles, and some babies born with HIV infected women. It can be also infected, as I said, by HIV. HIV can infected by an IV. So this is why the blood, before we do any intravenous injections of the blood, it need to be screened very carefully from any transmissible disease. In 2017, 36.9 million people live with HIV. Most of them, 10%, are under the age of 15. So far, the total death attributed to the HIV is over 35 million. This is actually an electronic micrograph of the HIV, of a cells infected by HIV. The graph in your left, it's a plot that showing the concentration of helper T cells in the blood per milliliters. And years in the x-axis. The T helper cells began at high concentrations and decline over the years. All line of HIV began at low concentration at 0 years and increased to higher concentration and then dips down again before the first half of the year. after which is steady increase. All those data are just approximate data anyway. The sooner we start the therapy for HEV, the better chance to destroy it. I mean, to not destroy your immune system. We're still working actually on developing an AIDS vaccines for those. The prevention for the moment is just use any preventions during any intercourse. Be aware of any intravenous drugs. Avoid any anorectal intercourse because those are the rectum is very thin and the affected T cells can enter the body here very easy. So let's conclude actually our lectures for today. I will post this recording shortly. Stay safe. That's again, it's Dr. Carver.